Now that we've learned about the structure and chemistry of the different types of lipids, it's time to talk about their functions, starting with fatty acids. Now, as we talked about previously, fats can be found in certain types of food, such as butter, oily food, and fat-rich meat, such as the red beef that we see here. And all of these are good examples of energy-rich sources of food. Now, the problem about this compared to sugar-rich food is that fatty food is an example of what we call long-term energy rather than the short-term energy provided by carbohydrates. Now, the simple distinction between short-term and long-term energy is that as long as your body has a repository of sugar, usually this is glycogen, your body will not use any of the long-term energy in the fats that you've stored in triglycerides. This is why for people that want to burn fat and lose a lot of weight quickly, they need to do rather strenuous and rather lengthy amounts of cardiovascular activity, because the length of exercise is needed in order to break down and consume all of your sugar reserves before it can start breaking down your fat reserves in order to burn off some of the fat that you've stored. On that note, let's talk about how bodies store fat. So there are two different ways that fatty acids can be stored for future use, and probably the more common or more specific in terms of fat storage is in triglyceride molecules like this one, which are stored in large fat storage cells that we call adipocytes. Now, even though adipocytes are informally called fat cells, fat storage is actually only one function that adipocytes have in a variety of different animals, which we can actually see taking place here. So if we look at the seal, the polar bear, and the beluga, we can see that all of these animals have rather large fat storage, and this is necessary because fat makes a very good type of insulation against stresses from the environment. One reason why these animals want large reserves of fat is that fat is a predominantly nonpolar or hydrophobic molecule, meaning that water cannot pass through a layer of fat very easily, which makes having large fat reserves very good for water retention. But most importantly, we would see seals, polar bears, and belugas in extremely north or extremely south environments where it is very, very cold. And the main reason why these animals require a lot of insulation is to prevent heat loss and trap the internal heat of their bodies inside to allow them to survive in very cold polar climates. Now, this sumo wrestler uses its fat to insulate against something completely different. Sumo wrestling is a very aggressive type of sport that's very easy to receive physical injury if you don't have insulation against some sort of physical trauma. So sumo wrestlers use their large fat reserves in order to protect their skeleton and all of their vital organs underneath. So a very different but no less important application of adipocytes and fat storage as a form of insulation. Now remember that triglycerides are only one method of storing fatty acids long term. The other method is by producing what we call phospholipids, which we recall are a combination of a phosphate head, which is negatively charged, and hydrocarbon tails, which are completely nonpolar because they're made entirely of carbon and hydrogen. And the primary location where we find phospholipids in any organism is in the cell membranes of that organism's cells. And cell membranes are simply the outer layer of a cell that prevents the inside contents of the cell from breaking out. Now, phospholipids have a very interesting structure compared to other lipids because they're not truly nonpolar. They're actually amphipathic, meaning that they have a polar head, because remember that the head of a phospholipid contains the negatively charged phosphate. Now, if we think of the contents inside of the cell and outside of the cell, both locations contain a large amount of water. So on the inside of the cell, we actually have the cytoplasm, which is the liquid part of the cell, which is made very, very, very heavily of water. And outside of every one of your cells, you actually find your blood, which is also mostly water, which means that water can be found both on the inside and the outside of your cell membrane. And because the phosphate heads are polar, that allows them to form hydrogen bonds with the water inside the cytoplasm and the water outside the cell found in your blood, thereby also preventing water from your cell from draining out. And the reason why this is true is because all of the non-polar layers of the phospholipids, all of the hydrocarbon tails, are all found on the inside of this structure, which is what we call a phospholipid bilayer, because there is an outside layer and an inside layer that face in the opposite directions. And the reason why this non-polar layer exists is, number one, the non-polar part cannot form hydrogen bonds with water, and number two, these non-polar parts are going to attract to each other because the only intermolecular interaction that non-polar molecules can make are dispersion forces, and those dispersion forces are going to hold the insides together and also prevent water from moving through the membrane, again, because water doesn't form hydrogen bonds with non-polar molecules. Now, despite the elegance of this structure here and the relative simplicity behind how this structure forms, there's actually a very huge problem with it, and that has to do with the fact that every phospholipid has a phosphate at the head, and that phosphate is negatively charged. So remember that phosphate has a charge of -3. Now, the problem with that is that if every single phosphate has a -3 charge and every single phospholipid has a phosphate at the head, that's going to mean that every single one of the phospholipids is going to be repelling each other and not want to stay close together. So how does this structure keep itself stabilized even if all the phosphates are constantly repelling each other? Well, in order to answer that, we need to take a look at the last family of lipids, that being the steroid family, and the prototypical example, cholesterol, which is this molecule here. And the job of cholesterol is to stabilize the structure of the cell membrane, and cholesterol can do this two ways. Now, cholesterol is similar to phospholipids in that the overwhelming majority of the molecule is nonpolar. The ring structure and this extra chain here only contain carbon and hydrogen. There is no electronegative electronegative atom, like oxygen or nitrogen, that can give this a strong negative or positive pole, which means that this section is nonpolar, which means that it fits very well on the inside of the phospholipid bilayer, because that means that it is going to be able to form dispersion forces with all of the hydrocarbon tails of the phospholipid. But we can see that cholesterol has a very conveniently placed OH group, and this hydrogen, because it's bonded to an oxygen, we know is going to have a positive pole. And that means that every single one of these cholesterol molecules is going to have that positive pole from the hydrogen, and we know that all positive charge is going to attract negative charge, including from the negative charge on the phosphates. So this one hydrogen in cholesterol is able to overcome the repulsion in the phosphate heads and hold them close together and prevent the phospholipids from flying off in every direction. And this is especially important when we have extreme temperature changes. So if the temperature gets hot, the entire structure of the membrane is going to move around faster. All of these phospholipids are going to be moving much faster, which means it's much easier for this membrane to fall apart. But because we have this positive pole and the strong dispersion forces in the nonpolar part, the cholesterol molecules can stabilize the structure of the membrane and keep this structure relatively solid and stop it from falling apart. Now, the final function of cholesterol actually has nothing to do with structure. Cholesterol is actually a base molecule that is modified in order to form another family of steroid-based chemicals, which are what we call chemical messenger molecules, although most people use the term hormone instead of chemical messengers. So, what are hormones? Well, hormones are simply molecules that are produced in one part of the body, transported through the bloodstream to carry a message to cells in a completely different part of their body. In other words, hormones allow cells in one part of the body to indirectly communicate and send messages to cells in a completely different part of the body. And the most well-known examples of these chemical messengers are, of course, the sex hormones that are produced by a specific gland in the brain called the pituitary gland. And during puberty, the sex hormones are released from the brain and travel to basically every part of the adult body and cause very radical structural and chemical and physiological changes within the body, including facial hair growth, pubic hair growth, breast growth in females, increased muscular mass in men, widening of the hips in women, and perhaps most importantly, the beginning of the menstrual cycle in women so that the period begins to happen. These are all caused by sex hormones, and as we can see that both of these are steroid-based in nature. Now that you've finished learning about the different functions and chemistry of lipid molecules, take a look at the two case studies that have been given here and the practice problems in order to solidify your understanding of the chemistry and functions of lipids inside of the human body.